Glycolysis is the breakdown of 6-carbon glucose molecules into 3-carbon 2-pyruvate molecules, 2 ATP molecules, 2 NADH molecules, and 2 water molecules, which the process take place in the cytoplasms of the cells. The enzyme hexokinase phosphorylate the glucose. Glucose is converted into glucose 6-phosphate. ATP is converted into ADP. The enzyme phosphoglucoisomerase isomerizes glucose 6-phosphate into its isomer, which is fructose 6-phosphate. The enzyme phosphofructokinase uses another ATP molecule to transfer a phosphate group to fructose 6-phosphate to form fructose 1,6-bisphosphate. 2 ATP molecules have been used so far. The enzyme aldolase splits fructose 1,6-bisphosphate into glyceraldehyde 3-phosphates and dihydroacetone phosphates, which they are isomers of each other. The enzyme triose phosphate isomerase rapidly converts dihydroacetone phosphates into glyceraldehyde 3-phosphates, which glyceraldehyde 3-phosphates is the substrate used in next step of glycolysis. The enzyme glyceraldehyde 3-phosphate dehydrogenase dehydrogenate glyceraldehyde 3-phosphate by transferring its hydrogen molecule to 2 NAD+ to form 2 NADH and 2 hydrogen ion. At the same time, two phosphates are added to form 1,3-bisphosphoglycerate. The enzyme phosphoglycerate kinase converts 1,3-bisphosphoglycerates into 3-phosphoglycerates. These reactions use 2 ATP. The enzyme phosphoglycerates mutase relocate the phosphates of 3-phosphoglycerates from the third to second carbons to form 2 phosphoglycerates. The enzyme enolase removes 2 water molecules from 2 phosphoglycerates to form phosphoenolpyruvate. The enzyme pyruvate kinase converts phosphoenolpyruvate into pyruvate. 2 molecules of water and 2 molecules of pyruvates are formed, which are then used in the Krebs cycles. Explain the step of Krebs cycle. In the first step of Krebs cycle, acetyl-CoA joins with 4-carbon molecule, which is oxaloacetate, releasing the CoA group and forming citrate. In second step, citrate is converted into isocitrate. In this reaction, a water molecule is removed from the citric acid and put back in another location. In the third step, isocitrate is oxidized and release a molecule of carbon dioxide, leaving behind a 5-carbon molecule, which is alpha-ketoglutarate. During this step, NAD+ is reduced to form NADH. The enzyme isocitrate dehydrogenase, which catalyze the speed of reaction, are used. Next, the alpha-ketoglutarate is oxidized, carbon dioxide is removed, and coenzyme A is added to form succinyl-CoA. During this oxidation, NAD+ is reduced to NADH plus H+. The enzyme that catalyzes this reaction is alpha-ketoglutarate dehydrogenase. Next, the CoA is removed from succinyl-CoA to produce succinate, and the energy is used to make GTP from GDP by substrate level phosphorylation. GTP can then be used to make ATP. After that, succinate is oxidized to fumarate. During this oxidation, FAD is reduced to FADH2. The enzyme used is succinate dehydrogenase, which catalyzes the removal of 2 hydrogen from the succinate. In the step 7, water is added to fumarate, converting it into malate. In the last step, malate is oxidized to produce oxaloacetate. During this oxidation, NAD+ is reduced to NADH plus H+. Thank you.
talk about electron transport system. In the electron transport system, NADH and FADH2 are used to produce ATP in structures called cytochromes. The energy released by electrons is captured as ATP by the ADP molecules. How the electron transport chain works: when NADH that is produced during Krebs cycle approaches to complex 1, NADH gives up its protons and electrons to form NAD+. During this process, it donates electrons to complex 1. The complex 1 has the energy to pump proton from the mitochondrial matrix to the intermembrane space. NADH enters the electron transport chain yielding 3 ATP per NADH. It pumps more and more protons from the mitochondrial matrix to the intermembrane space. This will create a proton gradient. Complex 1 then passes its electron to coenzyme Q. FADH2 comes along and approaches to complex 2. FADH2 gives up electron and turns into FAD. However, complex 2 cannot pump protons from mitochondrial matrix into the membrane space. The electron then passes to coenzyme Q. FADH2 enters at coenzyme Q, producing only 2 ATP per FADH2. NADH and FADH2 carry protons and electrons to the electron transport chain located in the membrane. The electrons are then passes to complex 3, then to complex 4. The energy from the transfer of electrons along the chain transports protons across the membrane and creates an electrochemical gradient. The final electron acceptor is oxygen. The electrons are then passes to oxygen, which splits into 2 oxygen ions. The protons are added, creating 2 water molecules. In conclusion, 2 protons, 2 electrons and half of an oxygen molecule combine to form water. As the accumulating protons follow the electrochemical gradient back across the membrane through ATP synthase complex, the movement of the proton provides energy for synthesizing ATP from ADP and phosphate, as protons will flow from a higher concentration gradient to a lower concentration gradient, which is from the intermembrane space to the mitochondrial matrix. The gradient exchange of hydrogen ions across the inner mitochondrial membrane is known as oxidative phosphorylation. Since oxygen is the final electron acceptor, the process is called aerobic respiration.